Good morning, Dr. Cummings, Dr. Hendrix. Thank you so much for joining me today to talk about your upcoming session at AAIC. I'd love to just get started. Dr. Cummings, if you could give us a quick little bit of your background and how you joined Dr. Hendrix on this project.
Great. Thank you, Marcy. I'm really looking forward to this event at AAIC. It's going to be a fantastic review of the history of 25 years. Unbelievable that it's 25 years. So I'm Jeff Cummings, really happy to be working with Suzanne on this program. I am a neurologist. I have a deep interest in clinical trials and drug development for neurodegenerative disease and Alzheimer's disease. I direct the Center for Transformative Neuroscience at the University of Nevada, Las Vegas.
Outstanding. Thank you for joining us. And Dr. Hendrix?
Suzanne Hendrix, and I've worked in clinical trials for 30 years, over 30 years now, and in Alzheimer's for over 20 years. I'm super excited about this session too. Really glad that Jeff was willing to work with me on this, to be one of the co-moderators with me. And I've reached out to several really prominent people in the field who are going to help us give an overview of the last 25 years of the field, everything that we've learned. And we've come so far. There's so many things we've learned. And then also looking forward to what's going to happen over the next 25 years. And I think it'll be a really nice session. And for me, it'll be a good overview. I know I'm going to learn a lot from the presenters. I kind of picked people that I wanted to learn from and wanted to figure out, some of the things they know and get a little more insights into it. And the part that's really going to make it exciting is the interaction with our audience, getting questions from the audience and then seeing what comes up in those discussions. So yeah, I'm looking forward to it. And you can just call me Suzanne also.
Awesome. Well, thank you both. With that said, I'm going to go ahead and start you with some warm-up questions before your session in July, because I have a lot of background information I'm excited to learn about also. So I think let's start with the 10,000-foot question was, what has been the biggest surprise or breakthrough that has occurred in the past 25 years that you feel has moved Alzheimer's research forward from where we were to where we are today? Jeff, do you want to go first?
Yes, that's a great question. And unequivocally, the most important thing for my patients, which is what matters to me, is the approval of new therapies that actually change the disease course. This is so important. And there are many targets. People have criticized the emphasis on amyloid. That's because we won with amyloid. Now let's win with tau. Let's win with inflammation. Let's win with a lot of other targets. But we have broken through. We've created a new era of therapeutics with disease-targeted interventions in the amyloid cascade. And I think that's so important because it's important to my patients.
Outstanding. And Suzanne, from a research lens, what was the major breakthrough that you think happened in the last 25 years?
So it's interesting because it's in a way, it's the same thing that Jeff said, but it's the other side of that coin, which is that we finally developed approaches and analytic methods and outcome measures and interactions with the FDA that could get us to the point of getting approval of a disease-modifying therapy. The reason that I went into Alzheimer's disease and kind of stayed in that field after I had worked there at a specific company was for that reason, that it's very, very hard to see slow, subtle changes in early disease. And it's hard enough that even once you see them, you're not sure if they're real. And honestly, we're still fighting that battle in a lot of ways where people think the monoclonal antibodies don't make a big difference for patients. But the difference that they make is sort of the equivalent of prevention over treating symptoms late in disease. And that prevention in early subtle disease is so important to see how much different that is than a symptomatic benefit. And so I think we're still fighting that battle in a lot of ways, but just to have gotten far enough that we could see success when we had it is really important. And there's still quite a few people who don't even recognize that success. And it's because that subtle difference between early prevention or slowing subtle change versus big dramatic change late in disease. So I feel like we're still trying to fight that battle, but we've made substantial progress.
Well, following up on that, Jeff, what do you think has been the most significant advancement that has occurred in how we are diagnosing Alzheimer's disease over the past 25 years?
Thank you for that question, Marcy. As I was listening to Suzanne, I was thinking the importance of amyloid PET was pivotal because we suddenly understood, one, amyloid was present in the brain for a long time before people developed symptoms, and that opened the door to prevention. And then we also saw that we were overconfident in our ability to make a diagnosis of Alzheimer's disease accurately, and that we were putting people into Alzheimer trials who didn't have Alzheimer's disease. So we've cleaned that up with amyloid PET, and then the many great tests that followed, like p-tau217, that is a plasma measure that reflects the amyloid PET status. So I think the advances in biomarkers, which allows us to diagnose Alzheimer's disease, but also allows us to conduct trials with so much more precision, have reshaped the field of Alzheimer's disease clinical trial research.
Outstanding. And Suzanne, following on that, I mean, I think one of the pathways to success always requires failures, right? We learn so much along the way. So can you describe for us a failure, if you will, that happened in Alzheimer's research that ultimately paved the way for the success of where we are right now and where we even hope to go from here?
Yeah, certainly. So again, following up on Jeff's comment, I think one of the most important things that we've done is identify the patients who have the amyloid pathology, being able to treat them with amyloid targeted agents and see a treatment effect there. And being able to very clearly diagnose that group is really important. And that's what allowed us to have success. But I think that there's another important side to that too, which is that now we have a lot of people who clinically look like they have Alzheimer's disease, but are not part of those that have amyloid yet. And so then the question is, what happens to them? Where do they go? How do we treat them? Which mechanisms are going to work with them, and which biomarkers are going to identify them? And over the next, hopefully soon, 5 years or so, but certainly over the next 25, there will be a lot of changes in terms of how we define that. It may be new definitions of the disease. It may be subtypes of Alzheimer's. It may be, something totally different. It may be something related to alpha-synuclein, and maybe it's some combination type of patient who has Alzheimer's but has some of the pathology related to some other diseases. Maybe it's some of the vascular causes and the mixed dementias. And I think there will be mechanisms that also target those types of treatments. And a lot of what we've done in the Alzheimer's space will lay the groundwork for that, where we define the population using really clean biomarkers, and then we measure the outcomes very precisely in early disease in a very, very homogeneous patient population where we're actually able to see those treatment effects.
Well, I think one of the biggest things that, you guys both touched on is the complexity, right, of Alzheimer's disease and personalized medicine, and understanding that there's so many pathways and different bodies respond differently in different comorbidities and things. So considering the extreme complexity of the risk and protective factors that we now understand through Alzheimer's research, what are some of the major hurdles in figuring out how they interact with each other? And how can that knowledge lead to more personalized and effective prevention strategies, treatments, and how is that articulated through the research?
I
John,
can
you want to go first?
Yeah.
One of the great hurdles right now is that we do not have all of the biomarkers that we need. We have reasonable biomarkers for amyloid, different forms of amyloid. We have biomarkers for tau and different forms of tau, but TDP-43 is frequently present in the brain, and we have no biomarker of that. Synuclein is frequently present in the Alzheimer brain. We have no biomarker of that. There's a CSF seed amplification assay that looks pretty good, but it's not really practical or widely used yet. We don't have very good plasma biomarkers of inflammation, and that's one of the primary processes of Alzheimer's disease. So I think in terms of the hurdles, it is the ability to see the co-pathologies that we know are there because we can see them at autopsy. We want to prevent them. We want to understand whether they are limiting the treatment benefit we can see with anti-amyloid therapies. We want to see whether we can do combination treatments with a TDP-43 drug and a monoclonal antibody, for example. These are the questions that must be answered, but we can't do it until we have a biomarker. So I see the identification of those biomarkers as a major challenge and a major goal for the field right now.
Outstanding. And Suzanne?
Yeah, from a statistical standpoint, I think a lot about interactions with medical history or comorbidities. And even when we don't have biomarkers to measure them, we kind of have some indication of the types of people who may have them, at least in our trials, based on some of those medical history reports, medications that they're on, things like that. It's been really interesting because there are publications that show that those comorbidities are good predictors of the patients who will both benefit the most and who will have the biggest safety risk in many cases. And there are publications that show that people who have, for instance, higher blood pressure are more likely to have ARIA risk with the monoclonal antibodies. But in a lot of cases, those comorbidities are the things that can be improved the most with treatment. And the reason is that everything gets worse when you have Alzheimer's symptoms. You forget to take your medication, you're not managing all of your other comorbidities as well. All these kinds of things are what really end up costing a lot of money and reducing the quality of life for the patients with these comorbidities. And so we see really substantial benefit with monoclonal antibodies, partly because these comorbidities are reduced. And so it's really a complicated interaction where you need to factor it in on the safety side and also on the efficacy side. And if we can be more explicit about it because we, get access to biomarkers that can give us better insights, then we can do it more proactively and do it in a way where those safety risks are minimized, but we still get the additional benefit that we might obtain in the patients who really need it and who hopefully won't be excluded from the clinical trials, because in some ways the clinical trial populations focus on a cleaner group. And once you go out to the community, there's a lot more comorbidities, and that's where the real big benefit will happen. So it might be that the benefit in the community could actually be a lot bigger than we see in clinical trials because of that broader group who can really have that benefit across all the comorbidities as well as for just the main core Alzheimer's disease symptoms.
And speaking of clinical trials, do you see a space where we're going to start to see the ages of enrollment in clinical trials start to reduce, thinking through that, risk perspective and the concept that change is already happening in the brain, 20 years often before symptoms are developing? Do you think we're going to start to see clinical trials that are going to be looking at individuals earlier and following them or in a longitudinal pathway, especially folks who have like the ApoE4 lipoprotein?
Let me jump in on this one first, if you don't mind, Jeff. This is one where I think it's sort of this trade-off, and we've talked about this a lot in the Alzheimer's space in terms of study design, that you can either study the disease when it's measurable or when it's changeable. Can you intervene early enough to make a difference and late enough that you can measure that difference? And for proof of concept studies that are really small sample sizes, it's very hard to do both. If you're a big company and you're in phase 3, you can just put more patients into the study and you can measure really slow, subtle, early change. But if you're a small company and you're trying to get proof of concept, you need to measure patients that have enough decline. And sometimes when you go to those earlier, younger patient populations, the decline isn't quite as steep and you're not able to see the treatment effects that you need to. And so there will always be a little bit of a disconnect between the population you need to study in a proof of concept small trial, and then the group you would study in a phase 3 that might be kind of intermediate, and then the group that will eventually be treated out in the community. And it's important to make those decisions carefully, because otherwise you can miss proof of concept when you truly have a treatment that works. So you don't want to try to go too broad too soon, or you might miss your chance for success.
Understood. Jeff, do you have anything to add to that?
I would just add that an important part of this dialogue is about biomarkers and their regulatory status, so that a trial could have a positive outcome of a delay in biomarker maturation if the regulatory agent had enough agency had enough confidence that that biomarker would eventually predict clinical benefit. And we're in the process now of accruing this kind of information which would allow us to have that kind of confidence. So I think there are a lot of moving pieces here, but two of them are the regulatory part of this and the biomarker part of it.
Understood.
Yeah, one thing I want to add to that, the biomarker part is interesting, because at first you can have a biomarker that could be an accelerated approval biomarker where you get full approval, but it's accelerated approval. You still need to confirm it with a clinical trial based on, so you do the study based on the biomarker, you confirm it with a clinical trial that then shows a clinical benefit. But the second option is really the direction that we should be trying to go, and we obviously are trying to go, which is where you have a biomarker that's established well enough as a surrogate that you get full approval without any kind of conditions. And it's not accelerated approval, but it's actually full approval based on a fully established surrogate. And so getting our biomarkers to the point of surrogacy is really critical, because if we can get full approval using biomarkers, that allows us to be studying patients early. It allows us to be in the stage where we're making the biggest difference and see the biggest benefit. And so that is the direction that we want to be going. And maybe an intermediate step is having a biomarker that could be accelerated approval outcome, and then moving it to where it could be a standalone surrogate and be able to be full approval, have full approval based on that outcome.
So wrapping around that, I know, one of the confounding factors in studies is always people's lifestyle. But in Alzheimer's, lifestyle is important and ties to risk. So what's one of the biggest unsolved questions regarding the impact of lifestyle, such as sleep, nutrition, physical activity, and stress management on Alzheimer's risk and progression? And why is it so challenging to unravel and to account for in the studies?
I'll let you go first, Jeff.
Oh, let Jeff go. Okay.
The importance of lifestyle is just now being recognized for clinical trials, and we have never had good measures of lifestyle. We do not typically assess diet. We do not typically assess the amount of exercise someone had at different points in their life. We don't know what are the reserve and resilience behaviors that have gone into that individual, and to what extent are they accounting for heterogeneity that we're observing in the clinical trial outcomes? So we need to develop those measures. With the Fingers trial, for example, we're really seeing a global emphasis on lifestyle intervention. We need to know much more about dosing and how one considers that, and how one ensures adherence to these lifestyle interventions. But I think it's an area of really terrific promise, and I think it's something that we should all be doing, right? We all want a better brain span that will allow us to live a full, cognitively healthy life, and I would see that as the background into which our medications are introduced when lifestyle is insufficient.
Understood. Suzanne, from the
Yes, I totally agree with that. And I think the finger studies have done an amazing job of moving that field forward. I think one of the really big challenges in that area is that the number of different lifestyle and nutritional interventions that you could do is enormous, and the way you define them in terms of dose and timing and everything is just so vast that trying to get a parallel group that has a true control that could, be a sham or a placebo kind of group for each intervention is nearly impossible. And so what that means is that when you're studying them in a clinical trial, you have to do a simplified version of it that you can have a surrogate or a comparable control group or placebo that can correspond to it. And it's interesting because even with that simplified approach, we're still seeing pretty dramatic differences and seeing pretty dramatic, effects in the finger trials and similar studies like that. But then that still doesn't tie all the way back to what would happen if we were doing multiple things together and more complicated combinations than they're doing in some of those trials, where we can't necessarily do a true placebo that goes along with it. And then you have the added complexity that the people who are more compliant with it often are different to begin with anyway. Their genetics may be different. They may have had more, intermediate or more early life healthy choices. And so a lot of those things end up confounding things. So if you can't do a randomized controlled trial, it's very tricky. And even once you do, the compliance can differ based on different patient characteristics. And those characteristics are very hard to capture and very hard to carry forward. But I certainly agree that everyone should be making these kinds of lifestyle changes. We should be able to see drug effects on top of them. It will be easier to see drug effects without them, which is an interesting concept. And if we see those drug effects without them, and then people also have those interventions, we may very well get interactions and positive synergy where we get an even better drug response because it's on top of lifestyle and nutritional changes. And I think the research that's going on right now in that area is really strong and going to be really helpful, should be combined with the drug approaches that we have that are successful as well.
So, Jeff, I'd love to know, since you're, a practicing physician, what role do you see patients and the patient advocacy groups playing in driving the future of research design and drug discoveries for Alzheimer's disease? I know a lot of patient advocacy groups are trying to get, lend their voice, participate in trials, et cetera. So, what kind of roles can the families and the patients do to help this research move forward?
Thank you for that question, Marcy. It's just so important. It's kind of a paradox that we have developed therapies for patients with so little input from patients. What do they want? How much do we know about what they want? How can we quantitate what they want? How can we know whether we are achieving what they want? But I am seeing, both at the FDA level, where they have patient-focused drug development guidance now, and at the trial level of involvement of individuals in the community who are either caregivers or very early patients or people who are at risk, known to be amyloid positive, for example, and getting their opinion and taking it seriously and letting them help guide how to construct these trials. And I think very importantly, how to minimize the burden of the trial. It's one thing to say, well, we're going to have patient-centered outcomes, and it's another way it's another thing to think about, well, how far is it for me to drive? Could this be remote rather than in clinic? How many scans do I really have to have in order to understand this drug? And I think those are questions we should be asking of all of our trials. We want more trials, faster trials, more definitive trials to get more drugs to patients. And that requires us to involve the patients. Nothing about us without us is what they say.
Outstanding. And Suzanne, following up on that, from the research design perspective, what are steps that you guys are taking to try to involve patients and families more in that process and reduce some of those barriers to access to participating?
From the statistical side, we don't have a lot of direct involvement with the recruiting, with the patient interactions. But I think the thing that we're working on that really is very patient-centric is this idea of saving people time and disease. Whenever people do surveys of patients, surveys of families, and they ask them what's important to them, there's always words in there like prolong or delay or maintain. And all of those words really mean saving people time in their mildest stages so that they can stay in those mildest stages longer, move to the worst stages as long as possible from now, really prolonging the healthier stages of disease and keeping them where they are as long as possible. And I think it's interesting because sometimes when people do surveys of patients and ask them what's important to them, they focus on symptoms and they say which symptoms are important to you. And in reality, this is a whole disease and the whole thing is progressive. And the symptoms kind of move together and they start happening, together. And when you do these surveys and these questions and you see all of those words like maintain and delay and continue to be able to do this or that, those kinds of wording really say the whole disease is important to me. And there might be specific symptoms that are important one day and different symptoms another day, but more critical is sort of that underlying disease progress and being able to slow it down. And that comes back to Jeff's earlier comment about how meaningful the breakthrough of being able to have disease modifying therapies approved really is to the field, because that disease modification is very different than symptomatic benefit. And we need to think about it differently and realize that that is a much more inherent benefit to patients and their families than specific symptom targeting.
So I'm also curious, the payer side of things always comes into this. So, we have biomarkers that have been approved. Now we have drugs that are available, but there always is the challenge of having the actual implementation, of healthcare and the paying for those treatment services matching up with the available science. So, Jeff, how are you seeing the world of payers, and are they embracing these biomarkers? Are they embracing the treatments? How are we going to make sure that the science that's coming forward and the research and the breakthroughs that are happening are actually going to get into the lives of families so that they're going to be paid for by health insurance companies?
very important part of the availability of new drugs is that there is payment for them. And we're fortunate in the United States that CMS is paying for a large portion of the cost of monoclonal antibody therapy, and they are paying for the cost of the amyloid scan that establishes the presence of amyloid in the brain. Now, that will probably shift towards a blood test now that a new blood test has been approved. But in many countries, for example, just last week in the UK, NICE, which is their funding organization, decided not to fund donanemab and lecanemab. Well, that means that many patients will not be able to be treated. Or maybe when it's approved in 2 years, many of those patients will already be beyond the window of being able to be helped by these drugs. So we are fortunate right now that CMS has stepped up and has said, yes, we're going to pay for these drugs. The evidence is sufficient, and we hope that this can set a global standard.
So one thing I want to mention is that the misunderstanding that people have around disease-modifying therapies really being very different than symptomatic ones, and this idea that we can evaluate disease-modifying therapies based on whether or not there's a point difference that's clinically meaningful at a specific early time point. That same misunderstanding carries over to the pharmacoeconomic modeling that's used for disease-modifying therapies. We've done quite a bit of work around pharmacoeconomic modeling of Alzheimer's disease with these monoclonal antibodies. And what you see is that when a treatment is given early and it prolongs an early stage of disease, and it's with a disease-modifying therapy where the effect is maintained, and we have evidence that it's maintained by an off-treatment phase where it's already maintained, you actually get a benefit for the rest of your life. What that means is if you have 18 months of treatment and you save people 5 months of progression, every milestone of the disease is now 5 months later than it would have been even if they never take the treatment again after those 18 months. And if they continue taking it after those 18 months, there's evidence for lecanemab that the open label follow-on continues to aggregate benefit on top of that. And what that means, again, is that every single time you take the treatment, that benefit goes through the rest of your life. And if you factor in the cost savings of that through the rest of your life component, it's really very dramatic. If you just think of it as the benefit you have just today, then you're really underestimating how much these disease-modifying therapies really make a difference. And we've put that modeling side by side, and the biggest thing that modeling shows is that early treatment is much, much better than late treatment because you're slowing it down when it's at a very slowly progressive stage. You're maintaining that highly functional stage as long as possible, and then they drop off later in disease, and presumably there will be competing causes of death at that point, and you may never even get to the point that they have the worst symptoms within their lifetime. And if you can prolong the time until nursing care is needed, until nursing home placement, and prolong the time that they're able to live independently, the cost savings to those communities is going to be enormous. So my feeling is that CMS has made the best decision, and that paying for these things will pay for itself. And so it's not just a question of being willing to invest in these patients and give them the benefit. It's actually going to pay for itself in the long run. And I think if NICE saw some of that modeling and saw that potential benefit, that they would realize that even in a healthcare system where it's paid for by NICE, that they would also have those cost savings and it would pay for itself.
Understood. Thank you. What do you feel is the most promising direction in Alzheimer's research that you're excited about right now? What's happening right now that you feel like we're on the breakthrough of something big? Jeff, do you want to go first?
Yes. For me, biomarkers are the most promising breakthrough that we're seeing. And I say that because biomarkers help us diagnose, but they also help us with every single clinical trial. Every attempt we make to try to intervene is informed by a biomarker. And the idea that we can now see so many of these biomarkers in the plasma, that our assay technologies are getting better and better, that our understanding of the relationship among biomarkers is stronger and stronger. Our understanding of the relationship between biomarkers and cognitive and functional outcomes is getting stronger. All of these things are coming together, and those are the drug development tools that are going to help us as a field. We advance one drug at a time, but with biomarkers, we're advancing the field. And I just think that's so important and so exciting. I just can't wait to see what the next biomarker is.
Outstanding. Suzanne, how about you?
My initial thought when you asked the question didn't go toward the biomarkers, but I love Jeff's answer because it does, it's the rising tide that floats all boats. But what I was thinking of was I'm excited about immune and inflammatory approaches. I've seen quite a few treatments from different companies that have mechanisms that are tied to inflammatory or immune type approaches. And I'm excited about that as sort of there's amyloid, there's tau, and then I believe that those mechanisms will also be really important. And I think we'll start to get some breakthroughs in those areas. And I think many of those interventions are broad and end up approaching the disease from several different angles. And I think that's also going to come in with some of the the active immunization approaches where we might actually get more natural immunities being built that are maybe broader than just targeting amyloid or just targeting tau, but could potentially be broader and target multiple mechanisms there as well.
Outstanding. Now, thinking for your young audience who's might be attending or listening to you guys today, considering the challenges and the breakthroughs of the past 25 years, what is a piece of advice you would give to someone who's just starting their career in Alzheimer's research or Alzheimer's patient care? Jeff, do you want to go first?
Yes, I would say to the person starting, be passionate about your work. Be really into it. Let your whole spirit go into the work. Spend the time that's necessary. Make it happen. Understand it deeper and deeper. Interact with all the people you can. Bring it to life. These are the things that will allow you eventually to understand whether it's a target or a biomarker or a process, an assay, whatever it is that you're looking at. If you bring your passion to it, you have the ability to extend that to the rest of the world. And one of the things that's happened to me in my own career is I was doing something that I really was so interested in and working on it hard. And it just turned out there were a lot of people interested in it. And that was so rewarding, whether it was developing the neuropsychiatric inventory or doing our work on the pipeline. It just turns out what we were interested in had communicative value. People found it of interest. And I think for the young person, other people will find their passions of interest also.
Outstanding. Suzanne.
Yeah, I would say the same thing, but I would say it from a slightly different angle, which is just don't leave anything to chance. Like when you're doing research, when you're designing things, when you're planning things, don't just assume something's going to work out fine. Actually think through it and say, what is the very best decision we can make in this case? Don't leave any corner, unswept or any stone unturned. And when I talk about that, what I really mean is, think through what outcome measures should you really use? Which timing should you really be using for your visits? What do you know about how your treatment works and which population is going to work in the best? And if you get arrogant about how well you think your new intervention is going to work, and you think, you know what, it won't really matter how I design the study, this treatment is so great that it's going to be easy to see no matter what I do, then you get sloppy and it never works out as well as you want. Your treatment effect won't be quite as big as you want. And so every one of these details can make a difference. And you want to give your drug or your intervention the very best chance of success. So make sure you address every single consideration and be very clean about what you would do if you were just barely going to make success. What would you do to make sure you made it? Because our estimates right now are that up to 20% of Alzheimer's trials fail for methodologic reasons, even when the interventions actually have activity. And if it's as high as 20%, then that means those people who knew their treatments work still weren't able to see success in their trial. So be very careful about how you plan and design your trial and don't take anything for granted. Really make specific decisions, follow through on each of those decisions, and make sure you're not being sloppy in anything that you're doing or ignoring any specific aspects of the way you're planning your study. And if you do that and your treatment does work, then you'll be able to see it. But don't get arrogant and don't think that it's just going to work on its own.
Let me reflect on that one minute. I love that, Suzanne. And I've often said to myself, Jeff, don't believe everything you think. Uh,
Right?
and I try to make an increasingly clear distinction between what I know and what I think I know.
Mm-hmm.
What I know, I can use in a clinical trial and use that to plan. What I think I know has to have a bunch of contingencies around it. And so I find in my own career, still, I am struggling with this. What do I know and what do I think I know? And I think that's just such a valuable lesson for young people. And that is what you're stressing.
Yeah, and the things you think you know, see if you can verify them. See if there's data you can use to try to figure that out. See if your own data and your animal studies might help you. See if there's data from published clinical trials that could help inform it. If you think you know it, try to confirm it, or maybe you'll get evidence that you're wrong. How many times, I mean, at my stage of my career, there've been a lot of times when I was really sure of something and I was completely, totally wrong. So just don't get arrogant on a personal level, and also question everything. If you know it and you have evidence, hang on to it. If you think it and you're not sure, find the evidence. See if you can confirm. See if you can get closer to what the truth really is. Ask your colleagues. It's a great community. We have a lot of amazing people in this field. Ask them. See what you can learn from them. You can often find insights and learn so much more if you collaborate.
Yeah, what I'm hearing from you guys is kind of that concept of leaning into growth and always asking more questions and being a lifelong learner. I think that's a critical part of research and just success in life in general, right? So one big last question for each of you. If we have this conversation again in 25 more years, what do you think is going to be the one major breakthrough in treatment or prevention that you hope that we are celebrating and we're talking about with the same excitement that we're talking about these biomarkers and these treatments and this time-saving design that we're talking about right now? What do you hope is going to be that major breakthrough in 25 years?
I'll go first so Jeff can have the last word on this. I think I'm hopeful that it will be that we never get to the bad stages of disease, that we never see moderate disease again, that we never see severe disease again, that we're able to prolong those early stages and slow them down sufficiently, that people never get past that mild stage of Alzheimer's disease. We can just prolong it essentially indefinitely. That's my hope. And what that would mean is we can catch it early and then prolong those stages that are very subtle and very, a lot less impactful on patients and their families. And I'm hopeful that in the next 25 years, we'll be able to do that, and that we can look back and say it was a combination of the biomarkers, the lifestyle nutritional interventions, the disease modifying therapies added on to that, mechanisms that we didn't really understand, but were maybe pretty broad and could attack different aspects of disease, and then combination therapies added on top of lifestyle and nutrition. I think with all of that together, we'll be able to prolong the very early stages of this disease, and hopefully have that mild cognitive impairment stage maybe be the last stage anyone ever gets to, because we just slow it down so much that we never get to those later stages.
That would be outstanding. Jeff.
I have very little to add to that. That's a beautiful thought, Suzanne. Let's make Alzheimer's disease a memory, and then the idea of early intervention, prevention, and never having someone go beyond the MCI stage, I think, is just a beautiful way to think about success in this field.
Outstanding. So if somebody has questions for you all that they want to ask ahead of time, or they want to give you time to think about, how can they reach out to you? Should they connect with you on LinkedIn, or how can they get a hold of you in advance of the conference, or where can they find you at the conference?
I'm open to LinkedIn or email. If people want to send questions ahead of time, we can collect those and bring them with us to our session. Certainly at the time of the session, there will be a place where questions can be submitted. I hope people will think about what their questions are. We have a really good panel. We'll have a lot of good discussion, a lot of different perspectives. And if they think of questions ahead of time, I think we'll be able to have a really rich discussion. But I'm open to email, LinkedIn, any way they want to approach.
Outstanding.
I would just emphasize the importance of thinking about their questions ahead of time. I won't have time to answer a bunch of emails about this. So,
But yeah, no, I won't answer them. I'll bring them to the session. I'll
That
let
was
our
my
panel
hope.
answer them.
My hope was that you guys would be able to bring them with you to the session and help
right.
have the panel help consider
Yeah,
those.
we could say that if they send questions, we'll try to bring them with us. We will definitely bring them with us, and we'll try to address them.
Outstanding. Well, I want to thank you both for your time. I really appreciate it. It was an enlightening discussion, and I know this is going to be an amazing session. So just thank you both so much, and I wish you all the best with your upcoming presentation.
Great.
Thank you, Marcy.
Thank you.
Thank you for including me.